So we were talking about this, and I started into this pack. And so now, if you remember, I told you about the 1,000 Genomes Project, which happened about 10 years ago, and it was there was different phases and stuff, and we talked about this, and then we talked about how it looked at different parts of the world and different subpopulation groups and so forth. So now we ended last time on this slide, and so I'm going to start now on this slide. Okay, so this slide gives a summary of the 1,000 Genomes Project, and it gives a lot of the key numbers that we got on people, variation in variance between people. And the sort of center slide shows kind of the center part of the slide shows the key numbers for an individual person. And they're like I said, I would go so far as to say to memorize these numbers almost, but they're just very useful numbers to know. So there's numbers like how many SNPs does a person have, or how many variants does the average person have. It's just nice to know what that is. It's about 4 million SNPs, and the indels, it's maybe 600,000, and the SVs are maybe 2,000 and so forth, giving you about 4 million variants. And like I said, these are numbers from the past. I mean, they're changing a little bit now with new technology, particularly the structural variant number we expect to be much larger in a newly sequenced genome now, because people have much better sequencing technologies. But the SNP number is pretty stable, indel number fairly stable. And those are good numbers to just kind of commit to memory. And then we talked about the fact that most of the variants that we're sequencing when we sequence a person are common variants. That means those are variants that are shared between two different people. So if we go and sequence 2,500 people, like the 1,000 Genomes Project did, most of the time we're going to be resequencing these common variants. And actually, that was one of the key aspects of the project that was kind of their goal, to kind of create a catalog of common variants. And also they leveraged the fact that these variants were common to more accurately call them. So they did actually in this project what's called joint calling. So they called the variants a little differently than I described. They kind of simultaneously called them on all the people together, and that gave them extra statistical power. Whatever. And so then after they finished, they ended up with about 85 million SNPs. But the key thing is that in this particular counting, only about a quarter of those 85 million SNPs were common SNPs. Okay, so you can see now most of the time what the new SNPs that they're finding in the project are these rare SNPs and so forth. And then they had corresponding numbers for indels and structural variants. Okay, and that's the one of the key take-home things from the project. Now, we're going to also talk in a second about cancer genomes, and I just want to put that number in context. So if we look at a cancer genome, there's not as much of a sense of a typical number there, but I think the idea here is that the number in a cancer genome is much less. It's a few orders of magnitude less than in the germline genome. So your cancer genome has thousands or tens of thousands of variants, whereas your germline genome has millions of variants. An important number to keep in mind. And then another thing that's good to keep in mind is the coding genome. So people talk a lot about the exome, the coding regions. So the 4 million or so variants in a person in the germline turns into about 20,000 coding variants. So the number is not exactly 1% of 4 million, and that's because the coding region is under somewhat stronger selection, and there's a variety of different reasons for that. But about 20,000, and then the corresponding situation for cancer genome is about, say, if you had 5,000 variants, you have about 50 coding variants. And so those are just good numbers to keep in your head to kind of get a sense of the sort of scale of these things. Okay, so now I'm going to go forward and so now let me tell you a little bit more about rare and common variants. So we're not going to talk so much in this class about GWAS and things like that, but I do want to give you a sense of like what people do with common variants and kind of one of the big distinctions between rare and common variants. So one of the key aspects of common variants is they're shared. And so if you make up a 1,000 people and you look at their common variants, probably, if that cohort is from the same population group, a lot of people are going to share that. And if you looked at, say, people that have common diseases, the common diseases, things like heart disease, cancer, diabetes, that, a lot of the population have. A lot of the people who are positive for the disease are going to have the same variants that healthy people have. So often in GWAS, what you're looking at is you're looking at the differential frequency of certain common variants. And usually if a variant is common in the population, it can't have a very strong effect. I mean, otherwise it wouldn't be common. It wouldn't be allowed to rise to such high frequency. So often with common variants, these are the ones that we're using for genome-wide association studies. And we're looking at things like, in the disease population, there's a certain somewhat likelier chance you'll have the variant, but not that it's there or not, okay? Because that's usually not the case for common variants. Now, rare variants are a different matter. Rare variants, like I said, only occur maybe in one particular person in 1,000 or a small number of people. And so there, you often will find, for instance, if you're looking at a cohort, you might see something like this, where you'll see healthy people don't have a particular rare variant. Maybe they'll have other rare variants. And the disease people have, say, this rare variant or that rare variant. But the problem is that you can't get any good statistics for these, because these are really sort of like one-off things. Now, of course, if you imagine looking at a very large cohort, you could get better statistics. But, for the cohorts that people are looking at now, where they're looking at thousands of people and tens of thousands, it's usually hard to get good statistics. So often what they do for rare variants is they kind of aggregate them. They might do what's called a burden test. We won't get into this in detail, but they'll look at a region of the genome and they'll say, is this region of the genome, any place in the genome, does it have a rare variant? And that might be thought of as the burden on the region. So sometimes these things are, people use the term burden test. And often rare variants have bigger effects than common variants. And you can that sort of makes sense. I mean, if they're rare, they have a bigger effect. They might kill you, or they might have a very dramatic effect, for instance, giving you a rare disease or so forth. And what the idea is that they can have that big effect because if they were to have it in the entire population, it'd be weeded out in terms of selection. But if it's only in a small number of people, you can have a very large impact. And the extreme, of course, is somatic variants, which are almost unique, not beyond unique to an individual. They're almost unique to a particular tissue or cell. And sometimes these are extremely rare, of course, and they're potentially very, very deleterious. And this is, of course, what you have in cancer. Okay. So now let me tell you a little bit more about the numbers. I gave you a little bit of a sense of things. So this is just the overall summary numbers from 1,000 genomes. So there's the same numbers we looked at a second ago, the SNPs and so forth in people, the indels and so forth. But there's more things here. And I just wanted to, this is a little bit of a difficult table to jump into right away, but I'd still like to walk you through some of it. And I wanted to give you that simpler table to start with. So first of all, the people are broken into continental groups. So, for instance, there's African, here's European and so forth. And this is, the different Asian groups, East Asian, South Asian, and so forth. And you can see right off the bat, there tends to be more variation in the African population than the other populations. And this is just in general true for all types of variants. And then the other thing that we can look at down here is we can see for a lot of the variants, say encoding regions, we can ask how many of these variants are what's called loss of function variants. That means they knock out a gene or they'll have a very strong effect. That's kind of interesting. So for instance, let's look at the European population here. This number is 150. So what that means is that on average, a person in the European, if you compare two people in the European population, there's 150 genes knocked out in one person relative to another, okay? Now this is, you have to take into account the diploid nature of these variants. So each person really has two genomes. They have a diploid. So the number of homozygous versions of that 150 is much less, but it's still interesting that there's a lot of genes in people which, you can have loss of function variants in them and people are completely healthy. So you have, people will walk around and have a gene completely knocked out in their, in their body. And it's kind of an interesting thing. And people are very interested in loss of function genes in terms of developing drugs. And for instance, one of the recent drugs that was developed for heart disease related to this PCSK9 gene, this stemmed from looking at loss of function variants. And then here's a lot of other types of variants here. For instance, if you look at non-synonymous variants, these are the ones that change the amino acid versus synonymous ones that do. And so here what you're seeing is of the 20,000 or so variants in the exome or in the coding regions, about half of them change the amino acid. Okay, so there's about 10,000 amino acid changes in a person, and about the sort of complementary number don't really change the amino acids. And then there's other statistics, but these are, like I said, they're actually kind of useful statistics to look at, and that's why I'm walking you through this table. Another thing that we can look at too is for structural variants, we can look at the amount of the variants that we find relative to the detection technology. And this is a little bit technical, but we did go through all the different ways of finding out about these particular structural variants for read pair. We also looked at split read and so forth. And the key thing here is that these technologies work differentially for different lengths of variants. And so they have what's called ascertainment bias, and that's a very important aspect. So for instance, if you didn't look at one of those particular types of ways of calling structural variants, you would be biasing the collection of variants you have to maybe prefer shorter length or longer length and so forth. And so a big issue with structural variants isn't only detecting them, it's also trying to get some sense of an unbiased characterization of them so you can look at their length distribution and so forth. Okay, so that's all for 1,000 genomes, germline variants. Now let's look a little bit at cancer genomics. So last year there was another project that completed called PCAWG, Pan Cancer Analysis Working Group. And this project, actually kind of like 1,000 Genomes, was sort of led by the Sanger Center in England. And it really tried to get a comprehensive catalog of somatic variants that we have in cancer. And again, it's just useful to know a little bit about these numbers. So now this project, I'm just going to start with just the single nucleotide variants. This project looked at many different cancers. So for instance, here's all these different cancers here. You maybe wouldn't know all these, but there's a bone cancer, here's a blood cancer, and here's skin cancer. And then here's the number of variants. You notice that this is a log scale. And I think the key thing to realize here is that it varies dramatically between cancers. And this is why we can't really say in cancer you get this many variants. It's just varies over many orders of magnitude. So you can see here, there's some skin cancers, you might have 100,000 SNVs. And then there's some other cancers, for instance, these piloastrocytomas, which you might only have 100 variants. And so it's a really mixed bag for cancer. I said your typical cancer is maybe 5,000-10,000 variants. I think if you look in the middle here, you'll see, for instance, breast cancer, which is very common, that has about that type of number. And then notice also that these numbers vary. Each of the dots is a particular patient. So unlike the germline variants, where everyone pretty much there is at 4 million variants, these are even within a given cancer type. Some people have many more variants than others. And so cancer, it's a little bit different thing. And then here I just showed just more details. So here's like the number of indels, number of structural variants. And again, the numbers you can see are much smaller. Notice there is a lot of large structural variants in a lot of these cancers. And some of the cancers are characterized by having a large number of structural variants relative to the number of indels you have. For instance, so if you look at this particular cancer here, it has a lot of structural variants relative to its rank in the indels and so forth. And this one may be the opposite, just a little bit of what people have found. Let me just move this a little bit. So